Hello, everyone. My name is Anchita Sharma, and I am a doctoral student from the University of Paris-Saclay, and I want to show you how we used M13 phage to make a tunable delivery system for genetic circuits. Synthetic biologists have programmed bacteria to act as cellular computers, circuits that take in signals and process them the same way a logic gate would. Moon and colleagues, for example, layered multiple AND gates entirely within a single E. coli cell to build one of the most complex genetic programs assembled to date. But that's the pattern across the field. Most synthetic circuits, even the sophisticated ones like this, are still unicellular, one cell, one circuit, and no communication needed. The moment you want to build a multicellular circuit that is logic distributed across subpopulations, cells need a communication channel between them. Traditionally, that's when small molecules, which are the HSLs or the quorum sensing signals. They work, but they are low bandwidth, they don't give you high programmability, and you run out of orthogonal channels fast. So we use DNA instead. A DNA package can be specific, long, and because you're encoding in sequence rather than concentration, you get high bandwidth, high programmability, and channels that you can engineer to be orthogonal by design. So, picture a sender that wants to deliver a DNA package to a receiver cell. Our carrier for that package is M13 phage. M13 is ideal here because it's non-lytic. That means it secretes DNA continuously from a living cell rather than releasing everything in one burst when the cell dies. The DNA package itself sits on a phage mid, and we built 5 sender variants that use identical phage packing machinery but differ only in the origin of replication. This part sets how many copies of the phage mid are available inside the cell to be packaged. That one change is a huge lever. Per cell secretion rate varies almost 3,000-fold across the 5 sender just from swapping which origin the phage mid carries. But high output isn't free. The highest secretors tend to grow the worst because packaging and exporting DNA costs the cell resources. Two of our variants, P15A and P-Coli, break that pattern. They secrete a lot and grow well, making them strong choices when you need efficient broadcasting. So the senders are sending, but how do we know the receiver actually got the message? We made this testable. So the receiver cells continuously express GFP and they start out green. The package carries the DNA that encodes for the guide, that is CRISPR interference or CRISPR-I. This acts like a programmable off switch once it's inside the receiver. The CRISPR-I doesn't cut the DNA, it just blocks the GFP gene from being read. So once the GFP turns off, we know the delivery actually happened. GFP turning off here is a direct readout that the DNA message arrived and worked. We saw receivers switch off within hours, even at low sender to receiver ratios. So we confirmed this also under the microscope in real time, where senders and receivers were co-incubated on an agarose pad and imaged for 5 hours. You can watch the video and also the plot showing here the GFP versus time. So it shows the individual receiver cells lose green fluorescence as the message is arriving and the effect is taking place. The takeaway of this presentation, swapping one small genetic part, the origin, gives us a whole family of senders with tunable output, and we have a functional readout to confirm delivery. That's the foundation for building more complex, multi-signal communication channels between engineered bacterial populations. That's it from my side. To know more about this work, come and find me at the poster session at the LPM Phages 2026. See you there.